Our idea was to express 3 different proteins that are translated from an RNA sequence in 3 different reading frames. So this means that the sequences are present in 1 string, but shifted by 1 base, where all 3 then had to be optimized for the target organism. And none of the commercial software actually could be used for that, so our dry lab team developed their own evolutionary algorithm. To quality check those sequences, we used Splice AI to find the non-canonical splice sites, which are usually not found by other tools. And we also developed a large language model to search through previous teams' data to make the search more efficient and context dependent. So computation and AI were very important in our project that mainly focuses on synthetic biology. And that is also the case for our panelists, who come from diverse fields that are all connected by these two topics. Our first panelist is Professor Dr. Kristian Unger. He's an AI professor at the LMU Faculty of Medicine, where his group is a part of the Department of Radiation Oncology at the LMU University Hospital. Professor Unger's work emphasizes AI-driven predictive modeling from molecular multi-omics, histological, and clinical imaging data. So now I would like to welcome you to briefly present your work.
So thank you very much for having me here. First of all, I'd like to express that I'm really stunned. I mean, as I learned, most of you are from biology, and when I started studying biology, it was like flowers and bees. And now we are now talking about synthetic biology, and which now integrates so many disciplines, and not at least computational biology and AI methods. So, as I said, I'm a biologist, and during my PhD thesis, I was exposed to larger and more complex data than they could be managed and handled as by Excel. So I had to get my head into programming, and that was my start into computational biology. Now I ended up with being appointed as an AI professor at the LMU Clinicum, and my topic is AI-supported precision oncology. And to get you briefly into the topic is, so when you imagine you're a patient, for instance, with an oncological problem in your head and neck region, you get to be seen by the radiologists, by the surgeons, and by pathology. And all of them, they examine you, they generate data from you, and this can be summarized as clinical data. And those clinical data, they are then going to be used by the oncology physician. In reality, it's a team of physicians that go down a decision tree, so they follow a decision model and will get to a treatment, which typically consists of a series of surgery, radiotherapy, and chemotherapy. The problem we have in oncology is that, and this is real data from a head and neck cancer cohort that was treated with almost the same treatment, that the clinical outcome varies a lot across the patients. So at the x-axis you see the overall survival, and you see that there are patients on the left-hand side that do not profit a lot from the therapy, whereas it gets better walking more to the right side. And what I, together with many other researchers, what we are trying to do is we try to recognize patients with a suboptimal prognosis, understand the underlying biology, and to offer them a more efficient treatment. So we like to push this distribution from the left more to the right. And our general approach is that we use the clinical data as they are generated anyway, and we try to complement them with what we call omics data. So we try to grasp mostly completely the different molecular layers we can get from the cells, from the tumors, or from blood specimens, along with histology data and along with clinical imaging data, to feed an AI model that allows to predict the outcome of the patient and the underlying biology. And this then is supposed to get to an AI-supported treatment decision that hopefully then results in good treatment. To give you really shortly some examples, so this is a regression model that's used to predict the outcome in glioblastoma, the most aggressive brain tumor. It's a model that only uses 4 different microRNAs, and that allows us to assign patients individually to high-risk and low-risk groups. So the high-risk groups are the Kaplan-Meier curves on the bottom, and the low-risk groups are those more on the top. So we can predict by determining this 4 microRNA signature from tumor cells or from the blood plasma, whether a patient is likely to profit from the therapy or not. And this model is determinable from resected or biopsied tumor tissue or from pre-radiochemotherapy blood samples. Then we do explainable classification by convolutional neural networks fed by gene expression data. The trick here is that we transform gene expression data, which are vectors, into 2D images. These 2D images are fed into a convolutional neural network along with the classification information, and these networks then of course can classify, but they also can provide us information about the biological mechanism that is likely to be important for the classification decision. So we do biology-aware classification using gene expression data. Further, we now touching the field of histology-based AI modeling. That means that we use the tissue slides that are generated as part of the pathological diagnosis. We digitize those images, feed them to a vision transformer that was trained on many tens of thousands of digitized tissue slides, and gets to a rather low-dimensional feature matrix that then can be fed into a neural network to be trained to determine patient strata. And further, we even integrate our omics data, such as RNA-seq data. That could be any other molecular layer, but we start off with the RNA-seq data and combine them with computed tomography data. So we have paired radiotherapy planning CT images and RNA-seq data from, for instance, head and neck squamous cell carcinomas, and generate neural networks that are then able to determine clinically relevant strata. And that was a brief overview of what we are doing, what we did, and what we are planning to do. Thank you very much.
Thank you very much, Professor Unger, for this really interesting presentation of your work. So I think this was a great start into our topic. Our next speaker is going to be Professor Dr. Antonella Pizio. She's the Associate Professor for Chemoinformatics and Protein Modeling at TUM and head of the Molecular Modeling Working Group. Professor Pizio uses computational tools for developing the next generation methodology for food design, and also develops predictive models to support the development of new therapeutic approaches. And I'm really excited to welcome you to our panel.
Thank you so much. Perfect. Yes, welcome. So I'm by training a chemist. I thought that from biology, from chemistry, we all end up to it somehow with computers. And since I studied chemistry, then I did my PhD in computational drug discovery, and my postdoc in food. molecule discovery. This was the transition from drugs to food, and this what brought me to pricing in a Leibniz Institute. And since my 1 year, I joined TUM, and we were talking about code to life, and then the unit of everything that happens in life are molecules and proteins. And with computer, you can really have the advantage to work on any proteins that you want, right? So every shape and every function. And as we see here in this picture, there are just some example of case studies where we're working also different things. And you can work on food, drugs, viruses, bacteria. So it's always sequences of amino acids that get very different structure and very different functions. So this is why it's always difficult for me to say what we are working on, right? Because you can really work mostly on everything. But the main focus of our group, since we are in this Leibniz Institute, is on chemosensory receptors. So these belong to G protein coupled receptors. We have in our body 800 of proteins that all look like this picture. And they do everything because they are in the membrane, so mediate communication between outside and inside for everything. And everything, when we talk about external and inside, will be chemosensory, like taste, smell. So we have more than 400 receptors in our nose and our tongue that decide what we want to eat. And if now we have proteins and we have computer, so we can approach this interaction between the molecules that we taste and our receptor with two different approaches. We can look at the chemistry, so the molecules, and the field that works in this direction is cheminformatics. So molecules should be transformed into numbers, and then when we have this numerical representation, the computer can make the chemistry. And for proteins, these are a bit more complex chemicals. So usually we use protein modeling or this physics-based method that can be used to understand how this molecule get a shape and how they move. And this was what happened 10 years ago. So we tried to use also the statistical method, this physics-based method, and now we have to deal with this new concept, right? Everything is faster, we have better algorithms, and everything that is computational became AI. So I present just briefly 2 examples because I didn't want to, 2 examples, one from the cheminformatics and one from the modeling. So from the cheminformatics, it is borderline because it's about peptides. We have the one of the projects is to try to make predictor of bitter taste peptides. So cheese and many food products produce many of these bitter peptides. Bitter is not pleasant, but if you want to understand how to modify it, we have to understand what is this bitterness. So the idea is to, if you want to predict activity from the structure, you have this very old concept that you understand the structure-activity relationship. And this old concept, I don't know, there is something in my slide, now this statistics become AI, right? So we have algorithms, and this leads to the first question, but maybe we will have time to talk what is AI, right? So what we consider AI for making these predictions. And our idea was, so since AI is very good in language, and peptides at the end are chemical structure, but can be represented also as sequence of letters, as text. So we can use this very nice algorithm that work with text to decode this language. So we consider this protein language, this peptide language. So we use this graph neural network and create classifiers. So the good things of these classifiers is that they work, and they always work very well. But the other thing that is less highlighted is that it can be interpreted. So it's not a black box, right? So you can go to the layers and understand what happened to these letters. So for example, in this case that we have bitter peptides, we can understand what are the motifs that make a peptide bitter or not, and also which is the position of these peptides. because then if we go back to the biologist, this implies a structure. So if you add something at the beginning or at the end, it changes the interaction with the receptors. And what is always about modeling is, it's good if you can understand something, but it's also good if it works really. So if you can validate it, right? And we have a strong collaboration with food chemists that can synthesize the peptides, can validate our prediction with sensory panels, right? So this is one very easy case. The other complex case is when these peptides become proteins. This was anticipated, but I couldn't avoid to talk about AlphaFold in this context. Right? This is the biggest revolution of the last years. And why is the revolution? It's because we know that proteins are just getting a shape like we see in this video. So the folding of the protein really depends on the sequence and how the energy landscape of the proteins goes. So the shape of the protein depends from the fact that this is the best, the lowest energy conformation for this sequence. So it's very easy, the concept, but it's very difficult. It's very difficult to calculate this conformation. It's so difficult that there are hundreds of years of trying to do this with physics-based methods. So even if this is a physics process, it's difficult to calculate. So there is a competition that is called CAST competition, and each year the participants can propose new methods. And you see there are targets that are easier, proteins that are more difficult to predict. For the easy case, it was always easy. For the difficult case, to get a high accuracy, for the difficult case, we have this boost with AlphaFold. I usually, now I have AlphaFold in my teaching, and I usually always like to show the video of the AlphaFold tool. I don't know if you saw, I can recommend this video because it's a short video of their work during the competition and after the competition. And I like it, not for so much, we know that it's a breakthrough, but because behind this AlphaFold there is really a group of young scientists, not a big group, but it's very similar to what we are seeing here. And I think that this is the biggest revolution of AI that is in the next generation ends. So AlphaFold was defined as the most complex piece of AI. I don't want to explain, many of you already know. So I want just to highlight the idea behind. So the idea behind is that this folding that we show depends on the interaction between residues, amino acids that can be not close to each other. And with evolution, we know that proteins change sequence but do not change structure. So it means that this interaction will not change. So in theory, so if you add a contact between a positive charged residues and a negative charged residues, this interaction stays. So you have a change that is complementary in the two sides, right? So if you have lots of sequences, you can identify this interaction. So the beauty, the success is the algorithm, right, that can analyze this data, but the data is already encoded here. So it's the biological idea that at the end lead to the results. And just to conclude, of course we use AlphaFold. There are, it's good to know also how AlphaFold works to understand the performance, but this is the case of odorant receptors for which we didn't have a structure. So this is the AlphaFold model of 2022, and the structure was released in 2023, and I think you can see that they're exactly the same. So AlphaFold was able to do the structure of loops without anything played. So an initial structure is not necessarily the predicted structure again. And I don't want to take more of your time. I just wanted to highlight the potential that the applications will have for, in this case, for having structural activity relationship of the size, or for having structures available, because this can enrich the large scale analysis of the proteins. I just want to conclude with something about computational in general. So I have this transition from food to from drugs to food. There is not really transition, but in the food sector, it's very important at this moment to have a food transformation, right? So to change the source of food, but we still want that it is good and that we can get what we like and what we need. So there is a lot of research in the direction of computational gastronomy. So using AI to make recipes, so that you go back home and you ask Alexa, I want to eat something that is good, that is local, but it tastes good, which is a recipe for tonight, right? And there are, of course, for everything there are degrees. The thing that now we see what AI can do, maybe we are not that far, right? But the problem with data is, the first concept that what you have here, you get out, right? So if you don't have the information for Alexa to what is tasty, you cannot get out. And this is a big problem on food. So we need to create this knowledge. The other thing is the concept of the blind man and the elephant, right? So if you see part of your data, the data can also be good, but you don't get the picture. And for food, having the picture is important, right? And the other thing is that dealing with biological data, there are data and data, right? There is data and data. And biological data are specific data. And for this, we need also specific people that are trained to use AI for this data. And for this, I hope that I didn't take too much time, because it's kind of big.
Thank you very much, Professor Pizio. It was a really interesting, more of a chemoinformatics and structural biology approach to that. And our next speaker is going to bring another different approach to this. So we're excited for the ethics and sociology perspective on this topic from Maximilian Braun. He is a doctoral researcher at the Chair of Science and Technology Policy at TUM and a technology consultant for data analytics and AI. As part of his doctorate, he worked as a social scientist in the Responsible Robotics project to research the ethical, legal, and social aspects of robotics and AI in healthcare specifically. So we're really excited for your perspective on this topic.
Every time I enter buildings like these, I get confused what is the soap and what is the disinfectant on the bathroom. So that's every time I realize that I'm sort of foreign to this environment. But what I want to talk about briefly here in these 5 minutes that I have the impulse time for is the question of how to actually point to the society side of things. So I've seen that iGEM itself tries to tackle also the societal dimension of research and development. And so I'm bringing here an approach that was actually also practiced in a particular project, the Responsible Robotics project, which has ended last year. But the approach lives on, and it's called Embedded Ethics in Social Science and Robotics Research. And so this is basically a couple of slides and suggestions of how maybe also this kind of research could also help researchers in the fields of biology, synthetic biology, or chemistry and informatics, etc. to also consider these dimensions. So just to contextualize the work that we've done, so this was an interdisciplinary project from the outset. So you see a couple of different chairs and a couple of different people involved. So, and the approach is actually sort of a mixed methods approach from all of these sides. So the Responsible Robotics project was actually the first project to adopt this approach, the Embedded Ethics and Social Science approach. But there are a lot of other projects now still active who also work with it. And the best way to summarize what we've been doing in these 3 years that the project lasted is actually that we used a toolbox with different methods from the social sciences, from ethics, and also from legal analysis, to sort of delve into this topic with a more or less open mind and also see which kind of ethical challenges, social challenges, legal challenges emerge from the work that we see, basically. So the main building blocks, just to point out here briefly, of this approach are basically to integrate the ELSA questions, so that actually emerged from genomics back in the days. If you think about the Human Genome Project in the 90s, so this was actually where all this research was sort of pushed and also became more and more important. What we try to do is less sort of map out existing challenges and write a research proposal where we say these are the challenges we identified, we are going to tackle them, but rather sort of becoming a part of the field and also letting these things emerge bottom up. So what we actually did was spending a lot of time in the labs, a lot of time talking to people, a lot of time also inviting stakeholders from around the project to just get to know which concerns and which questions arise, basically. Just briefly introduce you to the context. So we did this in the Geriatronics project, which is a lighthouse initiative at TUM, which is also still ongoing. So what basically explains, in a nutshell, sort of what we did is that you see this, actually this slide is not as it should be, but you can see it on the right here. So that you have these two circles, right? So on the left, you have the field, basically, or what you would consider the field, where we did interviews, collaborations, workshops, and also focus groups to just get to know the many perspectives of these people and organizations that you see on the left. So legal experts, nursing experts, and also practitioner nurses, healthcare system suppliers, also providers of the technology, so commercial suppliers. So this was sort of the space of stakeholders and topics that we tried to encircle. Also teasing maybe your minds of what would be important stakeholders, maybe in your fields, that would come handy doing something like this. And just to do a couple of spotlights for the mini project that emerged from this bottom-up approach. So one thing was that we did a nursing training at the Red Cross Hospital in Munich, which had an integrated ethics module, so which combined practical hands-on experience provided to the nursing apprentices that we met there, but also having sort of dedicated discussion sections and also just deliberation on the ethical, social, and workshops that we did with the Lego serious play method. So that's just a playful method. You actually use Lego, so this is really about Lego. And if somebody of you gets the joy to ever work with focus groups, you usually have this dynamic that a lot of people participate, but in the end only 3 people are actively engaged and do the talk, right? And maybe the other 3 people are leaning back and don't say anything. This is actually a good method to evoke all, or to actually make all people talk, because they have to build little models. So here they build models of care robotics, still preserving sort of certain pillars for privacy and transparency as formulated in the GDPR. And also we hosted a project week. So this was the project week format at TUM, which is, I think, still a thing, if I recall correctly. So, and we also hope to host it again. So this was basically doing what we've done in an interdisciplinary setting. So providing the stage for students to as well sort of draft interdisciplinary research proposals, because we all know interdisciplinary work takes time. You have to understand the perspectives of each other, and usually you spend, if you have a 3 years project, first year just to line out a common understanding of certain challenges and topics. And this was the purpose of this intervention, sort of into the education side of things. Yeah, and then of course disciplinary publications are also a thing that I think I'll leave it with that, and yeah, looking forward to questions and discussions.
Thank you very much.
Yeah, that's yours.
Thank you very much for your perspective on the topic. And now we are going to be jumping a bit into neurosciences. So our next speaker is going to be Professor Dr. Wiktor Mlynarski. He is the professor at the Faculty of Biology at LMU and active in the field of computational neuroscience. His research group explores theoretical principles of neural computation with a particular focus on sensory systems, and also studies functions of neural systems and their organization. So,
our group is interested in understanding this meaning of computation from those two angles, right? Developing theories and studying data and everything that lies in between. And for the remaining minute, I'll just show you briefly, very briefly highlight some projects which lie along this continuum, right? From theories to data analysis, all coupled together by these ideas of probabilistic inference information theory. So on one end of theories for theory's sake, we can develop, and I'm putting them here just as icons. I'm not discussing what those things describe, right? But we can ask how nervous systems estimate properties of the world based on lossy representations of signals and stimuli, for example, sounds. There is a certain mathematical formalism one can write around here and solve it set partially on pencil and paper, partially using simulations. But the principles we identify here apply not just to biology, but also to artificial systems, right? So, for example, this question is related to how to form efficient data representations for dynamic inference, right? So this would apply to any system, artificial, biological, which try to solve this problem. Then we can make a step, right, on that spectrum towards actual biology, right, towards actual biological data, and ask if there are systems which follow these principles in biology, how would they look like, how would they behave? And theories help us to derive predictions, right? So these are predictions, whatever those lines mean, combination of lines mean, they describe somehow activity of neurons, in that case the primary visual cortex, right, visual region of your brain. And assumption here is that if the nervous system follows computational principles derived first, that this is what the activity should look like. And then, I wouldn't be showing you this if there are certain qualitative matches between those, right? I don't have a discussion what these matches, what similarity of these lines mean. Now, if it's a good computational theory, it should be substrate independent, right? It means that it should be applied by different systems regardless of what kind of signals they process.
Today. So I'd invite everyone to sit down and start with the panel discussion.
As you have seen in the presentations right now, AI tools have had already a transformative role in biology and are changing the way we produce and interact with this kind of data. So, to find more about the state of AI and computational methods in biology, let's dive straight into the discussion. First of all, why are you using AI and use these methods, and why can't the previous methods previously in biology, like decades ago, still be used? What about, we start with you, Professor Unger.
So that's...
I think you have to turn it on.
No?
Yes.
Okay, yeah, I mean, thank you for the question. It's a good one. I mean, actually, I would disagree. I don't think we have to say goodbye to the traditional computational biology or the traditional computational methods. I think they're kind of complementary. So when it's about AI and all the methodology associated with it, I think we should think about the advantages coming with that. For instance, when you want to generate a prediction model, a regression or pure mathematical based prediction model, you have to make a whole lot of assumptions, which you probably don't have to make with AI methods. So AI methods are much more flexible when it comes to assumptions you have to make. So that's, I think, one of the drastic advantages when it comes to AI. Plus, the recent developments when it comes to foundation models. So there's so much knowledge that can be gathered by data all around the world, basically, that then are summed up in a model, and this model then can be transferred and used by others. So that's the two advantages that jump into my head.
So from your perspective, it's like a synthesis of previous knowledge and methods accumulated that are now condensed into bigger AI integrated models, right?
Exactly, but this doesn't spare you, so using more modern technology, and this is a kind of a warning, it doesn't spare you from understanding the basic statistics, the basic mathematics, and the data as such. It's quite tempting using a data set and just plugging it into a framework that uses AI and not understanding really what the ins and the outs of the data are. So, as I said, I think it's a complementary story at all.
Mm-hmm. And could you give the microphone to Professor Pizio? Because you already mentioned in your talk that there is evolutionary data in systems like AlphaFold. Could you go into more detail about that? And maybe what's your opinion on this? Like how many of the methods you're using are like from decades ago, and how many of the methods you're using are more up to date, like let's say from 2020 and onward?
Yes, I mean, I think that the answer will seem simplistic, but so I think that it really depends on the question, and this is why I think that the key is to have a progressive knowledge, right, so that you can decide what to use.
And does that mean that the actual workload for scientists like you actually increases because you have to know all the previous methods, but also the new up-to-date methods? And as we all know, the developments are speeding up. Like just in the most recent years, there's been so many breakthroughs. Most people here in the audience also know about, or probably all of you know about ChatGPT and the impact it had on almost every aspect of life. And what do you think? Does it increase the workload to keep up with these new methods?
Yeah. No, I think that, I mean, it's also a challenge for the new generations, right? So, I mean, I have mentors that are older that say, it was so nice to go to the library and read the papers, but we could read few papers. And now we can read many papers, but we cannot read all papers, right? So we have to find strategies, and so it's not necessarily going to be easier having more possibilities. But also, you know, with the new generation, also the skills are different. I don't feel that I'm that old, but I feel that every student who comes that is younger than me is technologically better, right? So this is what I was saying with growing of skills in different directions. So.
What does Mr. Braun here think about that topic? Because you have worked on integrating multiple disciplines into this, and how hard is it to get everyone know the methods of the other areas and get usable results out of it in the end? Because if we are using these new technologies, it should bring some benefit instead of only increasing the workload, right?
Yeah, workload. In interdisciplinary collaborations, workload is always an important topic. Maybe just following along the lines of my previous speakers, I think it also often matters. So we often make this distinction between basic or applied research, right? The more I think you come to the applied side, I think also iGEM is targeting sort of the spillover into markets and to come up with new products, etc. The more you have to actually also talk about the constraints that the market actually poses to you. And in robotics research, we have a very strong inclination to actually talk already about products in a very early stage. So we talk about prototypes, we talk about how these things are used then in practice. And then actually new AI methodologies and also approaches are not that well received anymore because you need explicit knowledge about the behavior of a robot. So we also talk about safety in this regard. So while I think this was a nice input from in the last talk that we had about this data analysis versus theory, where we had sort of this explicit formulation of theories versus let's see what the data comes up with. And I think here you see already like the thresholds. So in an interdisciplinary project, if you have robotics approaches, where you just try out, mimic behavior of robotics through machine learning techniques, why not actually being able to explain them explicitly in some sort of a mathematical way is actually something that is not well received by the market in the end. And there I think a lot of let's say challenges already arise, which is the beneficial thing of interdisciplinary collaborations, of course, can then work out together. And I think the more, just to make this point, the more you come from this basic sort of more lab-oriented work to applied, maybe to eventually also creating products, the more actually, this holistic view on what you're doing becomes important, I think.
Was that the same before AI kind of integrated itself more into these applied methods? Or is it, has this changed over time? That recently with all these new technologies, robotics in your case, is this changing how much people have to understand about other fields? Because AI is, at least from my experience, really good at understanding patterns. So is it really important to still understand everything about these methods, or can you use it as kind of like a black box where you say, okay, you have certain inputs, you have certain outputs, and then you don't care about how it works, but instead you just make use of the technology to generate benefits?
There's a question.
Yeah, maybe to you, but also Professor Mlynarski.
Go ahead. There was robotics were mentioned. I wanted to go back to the first question, right? First discussion point about whether, what the new methods that we have right now can do that old methods couldn't. Because we are at this very interesting point right now where most of this novelty, there is this feeling of this transition, right? There is a precipice, right? Things are completely different now than they used to be. And it's true, right? As was said many times, many domains of society and of science are changing through these technologies, right? Yet this is novelty in technological development, not novelty in the idea domain, right? All of the ideas, fundamental ideas, and I'm not talking about technical adaptations of this, but fundamental ideas on which all those algorithms rely were developed in the around cybernetics era, right? So that's around mid-20th century, where people were thinking what might be, principles underlying control in biological organisms and in machines. And that led, to McCulloch and Pete's famous works on neural networks, first works on neural networks, and then Jerry Letwin, and then many others, right? That leading to Jan LeCun and Geoffrey Hinton and all these names, which are now household names, effectively, right? And each of those people would tell you that the developments right now, right, these are all new methods, right? These are all new technologies which beat old one by a lot, but in the idea domain, these are all manifestations of old ideas, right? And what I am interested in, right, and the part of community I'm representing here, I guess, is what are the new ideas, right? What are these foundational principles that we didn't think of yet, and perhaps in 100 years' time, they could potentially lead to even more of such changes.
Actually, I think that's a perfect transition to the next topic, which is why, if you say it's the same ideas from long before, why haven't they had the impact before, but only are arising now? So what has stopped these new these ideas from manifesting way earlier?
Well, I mean, maybe I can go, right? Because it's a fascinating question, history of science, right? And it's a mixture of many things, right? Mixture of the fact that ideas take time to mature, ideas take time to be accepted by the community, ideas need to arise in a certain context, right? I've seen some of the citation plots, whatever we think about citations as a measure of scientific quality, right? That's a separate discussion, right? But certain citations plot, they're for sure a measure of scientific popularity, right? Of certain foundational works from around the 80s, right? Where they were not cited zeros, and maybe 1, 2, blips around 0, and right now they're exponentially rising 40 years after, right? Which I guess is a quantitative manifestation of what the trends that underlie your question, right? And in history of machine learning and neural-inspired machine learning, there are many examples of it, right? There are many examples. The whole discussion about AI winters, right? Cycles of AI winters and summers, which many of you may have heard of, which are linked basically to misunderstandings and huge egos debating, right? Development of computer technology, right? What Geoffrey Hinton says, one of the anecdotes, Geoffrey Hinton anecdotes, right, is that he managed to make his algorithm 10 times faster, but over the time he made it 10 times faster, computers got 100 times faster, right? So he could run it with the previous one, right? So development of technology actually enables those algorithms to work in the real world. But I would like to also end, we're invited to be opinionated, right? So I want to end with an opinionated note, right? That for new ideas to be appreciated, right, there has to be intellectual atmosphere which values this, which values ideas without direct application in mind, right? Which values risky ideas, which values ideas which are not of immediate utility, right? And I am mildly worried, mildly concerned that this might be a trend which is, there might be some, I guess in German it says Gegenwinde here, right? So opposing winds, right?
Yeah. Actually, I would be very interested in the medical side here. So, what do you think are the factors that have held back these kind of technologies so long? Especially the medical domain is probably a little bit conservative. What's your view there?
I wouldn't put the other way around. I wouldn't say held back. I think they are now enabled, or again, differently put. I think the threshold really was lowered by AI technology. Or the accessibility. So if you think of applying any biocomputational method, 10 years ago, so you had to find this method. You had to find someone who you had to find an explanation for this. You had to get your head into this, and of course you had to program everything. So and of course you needed to have the environment for this. You had to have the computational framework for this. Now, what do you do now if you want to apply any method? First, you probably ask ChatGPT or any other foundation model. It will tell you, okay, you use this and that method, and basically you don't have to know how to program. It's enough to do meta programming. So you just provide your data frame or whatever and say, okay, do this and that with column X and column Y. And so basically everybody who is not from the domain can do things. And medics, again, and everybody coming from the medical field are not from the domain. I think actually it's really the accessibility that makes the difference. The accessibility enabled by new technology. That would be my answer to that.
What about Professor Di Pizio? Because you showed these the data points about the cusp competition. So what was the cause of this breakthrough over time? That now we have amazing models like AlphaFold that beat the previous methods by so much that they are now very or relatively reliable for real-world use cases.
Data. So because if you have models, right, we need the data, and I think that this, if I can jump to another point, open another concern, because going to popular science, like now there is machine learning of everything, right? You can always find patterns. You have data and you find something, right? But if you don't have the right data, this is the elephant story. If you look at something that is not complete, you get predictions that are, not applicable. And this is why I think that, a model is always a model, and the applicability is always needs to be validated and in a context, and the data should be, the driving force. So the AlphaFold success was the consequence of a lot of work on sequencing. So it's a revolution after another revolution. Without sequence, you cannot find pattern, right? And if you do the same in domains where you don't have data, I'm not sure you get the same. ChatGPT, again, we have lots of text. But if in English it's different than in German, right? So, I think that there should be also a responsibility of having the domain knowledge or where you can apply these new techniques.
That's actually, again, the perfect transition to the robotics field here. Because if you say data is the bottleneck, there has been quite recently a lot of advances made in that you don't necessarily need to collect the data from the natural world, but instead can generate synthetic data. And especially in robotics, there is quite a few companies who use simulations. One of the companies at the forefront of this is, for example, NVIDIA, who have huge robotic simulation, and then they apply the data generated from these simulations into robotics and to predict, for example, movements before applying them in the real world. What's your perspective on this? Will data, at least for the future, still be a bottleneck?
I think what we are currently seeing is probably, so this is what everybody suggests, right? So we have this phenomenon of model collapse. You have this thing of the internet is becoming more and more synthetic. So all of these these concerns that we're currently seeing is actually hinting towards this very problem. But I just want to make maybe another point. The worst thing you can do, taking a question and answering it. But I think language is always a concern, right? So the big question for me is always, when have we stopped? When did we stop? Because we now talk a lot about models, we talk about data, we talk about methods. when did we stop doing this in the public sphere, right? So we're talking about intelligence, we're talking about generative artificial intelligence, so we even cannot really differentiate if everybody takes into his or her mouth the word AI, what this actually means, what are we talking about? And you know, instead of talking about models making predictions in a multidimensional room, trying to get to the lowest point, we talk about, you know, generative AI being you know, the next big thing and coming around the edge sooner or later. And I think this is just something that maybe is a staple for all the comments that we've had about this aspect of responsibility. That also these hypes and these hype cycles that you've seen in AI, I think there was just a lot of over-promising, and also probably, which was a profitable thing for research as well, right? So that you actually get the resources for doing this, but we always get the aftermath, and also this sort of disillusion moment, which actually now also is a boomerang effect. So responsibility in that sense could also be, you know, talking about language. How do you actually represent this whole thing? And there are, of course, big actors out there who propose their vision about AI. But I think, you know, for us, if we talk about a vision of embodied AI in the form of robotics, what do nurses say, for example, about, you know, using robots there? How do they contribute maybe to this vision? Maybe they have other concerns. Maybe they should also be heard. So I think this also points to the importance of including different perspectives and different voices in this discussion, and also to shape these ideas that circle around in society of what is going on now with this big technology leap that we sort of anticipate.
You mentioned concerns. That's actually, again, the perfect transition to Professor Mlynarski, because there has been a company in the computational neuroscience field, but more on the application side. It's called Neuralink, a company from Elon Musk, and they have developed an implant that can somewhat read the human mind. At least that's what they want to sell you from the idea and what the media are promising. So what do you think? Is this an exaggeration and like some kind of illusion that's not really true? So, or on the other hand, is it actually possible to read the human mind as a computational neuroscientist? There, your perspective would be quite valuable.
That would be very disappointing to have a chance for such an interesting, engaging discussion and end up discussing to Elon Musk, you know, and especially given his recent shenanigans. You know, just to provide context, what Neuralink does is, in terms of scientific achievement, right, has been done many times by many laboratories working brain-computer interfaces, right? Including moving the cursors by either monkey or human patients, right? Well, the actual claim could be, and I'm not an expert in this, an implantation technology, which is also, you know, there's a lot of concern there, right? But, you know, your question was about reading the human mind, you know, so I think, let me take this as an interesting jumping off point, right? Because exactly, I think one of the things we want to discuss here is challenges, right? And in natural science, especially sciences which don't have yet well-defined theoretical frameworks, right, such as, I would say, biology or neuroscience, right, the most important challenge, I would say, is identification of the correct level of abstraction to interact with scientific problems, right? So, on one hand, we have this very big question, very abstract questions, like the one you just formulated. How do we understand human mind? How we cure cancer, right? How we feed the hungry or come up with drugs, right? They are very easy to state, but they are not scientific problems. They are just our like desires, right? On the other hand, we have the data, bits, ones and zeros, right? Mind-numbing sequences of measurement, right? And right now we can acquire more of them and faster, and it's great, right? But it's as concrete as it can get, right? And actual scientific understanding lands somewhere in between those things, right? I want to make a meaningful step towards the question, what does it even mean to understand intelligence? What does it even mean to read human mind? But I don't want to do it in the space of, you know, a beer discussion. I want to make it, you know, concrete, right? But again, still, so step in that direction, but a tangible, meaningful, quantitative scientific step. Right? And that means that I have to choose somehow, develop a style of thinking about the problem such that I land somewhere on that spectrum, right, from raw measurement to these huge life's questions. And I think, you know, working in is it computational neuroscience, theoretical biology, artificial intelligence, we stumble into these kinds of questions. What is intelligence, right? What are the and we sometimes tend to go around them, but how to address those, that I think is the most interesting of intellectual challenges here.
But would you say that it's possible to understand the human mind better with these kind of technologies, or is that a misconception where you would say, okay, it's over-promising from the AI field, like in previous AI hype cycles, where they say, okay, it's a huge breakthrough and so on, but it did show little application for everyday life, for example?
Yeah, so previous AI hype cycles, you know, the reason for these hype cycles are many and different, right? And it's a somewhat nuanced issue what it came to those hype cycles. I think what's fundamentally most important is that we are honest, right? We're honest about the methods we use, and we're honest about what kind of limitations they have. And to answer the question very directly, is it possible to understand them with those methods? Well, possibly, right, as with any method, right, that we used to have or will have in the future. Those methods will be superseded, right? Our grandchildren will be sitting here, will be laughing, you know, at those, you know, silly methods we had here. Or maybe, you know, maybe the principles again will be the same, right? But I think that question is not so much question of the method itself, right? But again, right, intellectual honesty and understanding what am I actually studying, what my method is revealing, what are its own limitations.
Actually, that's a good question for Professor Unger as well, because I would be interested, you mentioned some methods for explainable AI previously. So is it a potential tool where you say we can further our understanding of, for example, how cancer arises, or which kind of biological functions are impacted by cancer? Or is it more along the lines of a black box where you, even if you try to reach an explainable perspective, it's still giving such small tidbits that it's not really useful for understanding the deeper issue?
Actually, explainability is one of the requirements we have to fulfill when we talk about prediction models. So if you remember the projects I've introduced you to, and for microRNA signature model. So this is a regression model. So it's basically one of the most traditional methods you can think of when it comes to prediction. That you have a handful of molecules and you measure them, and you have then the coefficients associated with them, and from that you get a risk score, you split it and assign your patients to different groups. This is a really nice model. It works very good, but it doesn't tell us anything about the biology. That's the truth. I mean, it's microRNAs. Nobody, as far as I know, and I've talked to a few of RNA processing experts, they can tell you what a single microRNA does. And if you have 4 microRNAs, nobody can tell you what these do. Compared to AI model, which is a convolutional neural network, where you don't have to reduce the information you feed the model with. This model then at least has the potential to be explanatory about what is behind the prediction decision or the classification decision. So I don't know. It's somewhat contradictory, because everybody says, okay, or many people say, okay, AI, it's always a black box. I don't like AI because it's a black box. We don't understand what the black box does, and that's why we don't want it. That's true. I mean, nobody can understand a neural network. It's simply not possible. But you can, of course, and this is what all the explainability methods show, you can, of course, explain or at least point out what was important as an input to get to anything your AI model says. So this was a very long answer to a very short question, but that's what I think about this.
Yeah, Professor Pizio, quick question here. Do you think in the field of designing, for example, proteins, are there, does it work as promised? So you say, okay, you design the structure of a protein, then you can make inferences of how it actually works. So does that always work, or is this like, let's say, if somebody told you, okay, now that we can design proteins and so on and predict all the structure.
I think that, I mean, if we're talking about AlphaFold and modeling, I think that the biggest speed up was in protein design. So, I mean, I think that I linked to what was saying before. So one thing is to think on these big questions, and one thing is to move forward to this big question. I think that we have, I mean, in my perspective, we think of science is if you can add knowledge, right? And in this perspective, the more you can add, the closer you can get to the question. But this is why I was mentioning this thing of data and biological data, because it's so simple and so difficult that I don't think that we are, I mean, we can do the magic and say, okay, I want a sweet protein, and I click the button and I predict the activity and you can eat it, right? So I think that it's very good to have an accelerated science, but I mean, we have to train computational chemists, computational biologists that use the computer. So when we do the models, I mean, all the models are wrong, all the models can be useful, right? But when we use any model, we can predict big number, right? For my bitter predictor, you can give me thousands of peptides and I can make the prediction. But then we can test 10 of them, right? I mean, there is this limitation of the experimental work on the validation. So the one that I can add the knowledge are not hundreds, right? The one that I can say, okay, I found 10 more beta peptides. And this selection is not the computer. These are, I mean, in this case was the PhD student who selected. I always say that the validation was the model and Alexandra in this case, right? I mean, it's the person who used the intuition, who has the knowledge of how bitter compounds look like. And this, I think, is difficult to incorporate in the computer. So this is why I think that it will work if we also have a training of people that have the knowledge of the domain. And not everyone can do, you know, the predictor of bitter taste of cancer, right? So you have to know what is the field that you work with, because this is what will, you know, train and what will drive then at the end.
Is this limitation maybe on the sociology side? That we don't have enough skilled people, or that, for example, the really skilled people all go to the US because they have more attractive conditions, or all the big companies there? Do you think that's an issue here in Germany in particular?
I think this question is really clever because it points to the conditions of how we so we always operate in, you know, a space of confinements, of limitations, right? And I think that, yeah, which sort of make up also, which hugely contribute to the outcome of things, right? The thing that I was sort of reminded of of what you just said was basically the question of data availability also. So what kind of data are we talking about? And I think there is no case, probably not so much on the basic side of protein folding, etc., but anything that was related to social data that was collected to train models over the past decade, I'd say. They were always embedded misconceptions based on what that kind of data or what kind of social phenomena, so to speak, this data would represent in the end. And I think this is also a condition that we have to deal with when training models. So whatever data set you find probably was not customized or tailorized to the particular context of application or context of use. if you talk about social phenomena, and I think there are certain emblematic examples, right? So we had the issue with gender shades that were, FaceNet representing male whites foremost. Or I think even if you talk about medical data, like in radiology, do you have enough different images to represent population as it would be representative, and what does this representativeness mean in different contexts? For example, race is always this important thing you have to consider. I think in Brazil there are around 50 different boxes you can tick if you become a resident of Brazil for which race you actually belong to. It's a totally different conception as if it would be in the US, for example, which actually explicitly deals with these things, black, white, etc. That we also do not use that much in Germany. And I think it's just the conditions of where this kind of technology operates, and also how the information or the social conceptions about what the data should represent in the end also sort of have an impact of the output, right? And I think explainability here is also key to sort of find these correlations or interdependencies between the data that is inputted, and also awareness, of course. So we shouldn't use models in the assumption, but I don't think that we do it. But this is just something to keep in mind if we now have things like ChatGPT going wild. And I think we all probably are validators of ChatGPT right now, and we all know we couldn't validate it in many different cases. But yeah, just to keep in mind that the outputs that you receive are also shaped by the conditions and the social environment from which they originate, basically. Yeah.
Let's maybe rephrase the question in terms of accessibility, because you specifically mentioned that. Do we here in Germany, maybe even in the Munich area, have the accessibility to all the underlying data? To all the underlying data, or the talent, the people with the knowledge, or the computing infrastructure to actually make good models instead of biased models, for example, in your case? Do you think there is a big difference regionally, for example, between Germany and the US? And is that a big limitation if we want to apply these methods here as well? I mean, still you if you want.
Okay. So I do not work so much with data or training machine learning models. So I'd rather hand over the microphone maybe to have some actual experience on this one.
So, maybe I can say something about the medical field. Actually, so Munich, with regard to, I mean, there are two big universities. There is the Helmholtz Center. There is a really huge research environment, and particularly, I mean, if you think about the computational field, I think Munich is just an oasis, right? Also with regard to computational resources. It's probably unique in Germany, maybe even up with Berlin, but it's unique. When it comes to data and data availability, I don't think we have really a unique problem in Germany with having and generating those data. We have rather a political problem, and I don't talk about data protection. It's really political about the bringing those data together. For instance, when we talk about the cancer field, when we talk about cancer, there was a big effort in the US. Everybody who works with oncology data knows that. That's the TCGA, the Cancer Genome Atlas. And they took I think the most frequent 50 cancer entities, and they just collected every patient they could get from many hospitals, and they did every molecular layer, and they generated a big, huge data set everybody around the world uses. In Germany, we have the same potential, actually. So, but the problem is, for some reason, the big universities, they don't work together in a way that such an effort like TCGA in the US just exists. And this is a huge problem. I was talking about glioblastoma. Glioblastoma is very rare, luckily, but it's at the same time the most aggressive tumor, and it would be really cool to find something that would improve the treatment. the treatment basically is the same since 20 years. There was a small improvement some years ago, but still you will, in the median, you will die after one and a half years. There's no improvement at all. Now, as I am I'm one of the researchers dealing with glioblastoma, so the LMU university clinics, they see 80 patients a year, which is not a lot. You could easily double that by 2 universities working together, or by all the, say, DKTK universities working together. For some reason, it just doesn't happen. And I really see that there are political, strategic issues that prevent Germany of being as successful as it could be, as it could be, basically. And this is what I think about data availability and the issue with Germany versus probably other regions in the world.
Can I
Yes, of course.
So, you know, I'm not authoritative now to speak about accessibility computing resources or whatnot, or politics behind it, right? That's not something that is of my concern. Our needs are relatively small and they are met and we're all very happy, right? Nor do I, work in fields which require these very specific kinds of data, right? We have clinical data, for example, right? Our collaborations both in Munich and across Europe and the States are all rosy, right? So it all works. But the point I wanted to make, which, maybe it's what echoed in your question, set a kind of concern, right? Especially when one speaks to an audience of young people, right? Of people who are thinking what to do next, where to study, where to do research, what to work on. And you may be tempted, right, that we'll be choosing places, we should all go to Boston, right? Yeah, that's basically the bottom line, right? Or try to make all other places in the world like Boston, right? And I would think that would be a tremendous disadvantage, and it's also one of the, globalized science has infinitely many strengths and advantages, but it also has weaknesses, right? And one of them is trading exploration for exploitation, right? Look what happened to biology. Biology right now is 6 model organisms, or 7, right? 8 if you count humans, yeah? And we can all name them, right? C. elegans, Escherichia coli, right? What else, right? Mus musculus, right? Aptus novagus, whatever, right? And also all botany is just one plant right? Or 80% of it, right? There's this vast world out there which we don't study. Also a vast world of ideas and methods which we don't study if we try to recapitulate exactly what's in these, beacons of science. So speaking to what was mentioned before, that you always exist in some context, right? In some social-media context, some cultural context, some economical context. What makes science so hard anywhere is to learn things about the natural world, not given the ideal circumstances, but given circumstances you have, right? In the time you were born, in the place you were born, right? And to just, add to that, I very often think that having all of the available resources, very often, again, I'm speaking about, certain kinds of fields, right? In some cases, it is limiting, of course, right? I'm not dismissing legitimate concerns, right? But in many cases, having all available resources in those beacons, right, becomes also an excuse to do lazy science, right? It becomes a double excuse. In one place, you say, we cannot do good science because we are not like, MIT or HHMI or whatnot, right? And in those places, there is a reverse trend, right? Well, we are MIT or HHMI, whatever we do is going to be great, right? And that also happens, right? So discussion about resources or constraints should not discourage us from considering how could we best contribute to understanding nature in the context we're in.
Maybe to wrap this discussion up, I would like to have some input from your specific fields in what kind of advances you think AI methods will enable in the future that could have a really big impact on your field, and maybe not just on your field, but also for the general public, where they can notice it changing their lives in some way. Maybe let's go in this reverse order.
Reverse order, yeah. That gives me less time to think. Again, right, I come from the field where there is this kind of, what we call right now artificial intelligence or machine learning neural networks. There's kind of parallel, love-hate relationship, right? Where they're cousins. Those kids are cousins, right? They're closely related, and yet they hate each other very often, right? Yeah. And what I think of the, let me go this direction, right? There are many, of course. Let me go this direction. What I think about, for example, deep neural networks right now, right? Which are used broadly also as models for the nervous system, with varying degree of success. I think of them as a huge intellectual exercise, right? Because we have right now, first time in human history, we have this device, and this device does very well on many problems, also the problems that we humans or animals solve, and we can, neuroscientists dream. I can know every neuron and every synapse at every point in time, right? And it was mentioned before, I have no idea what it does, right? I don't even know what would it mean to understand it, even though I have all of that data, right? So this became a very useful intellectual exercise for us as a field to think, okay, guys, but so what would it actually mean to understand the system, right? How could we understand the system? And maybe even if those artificial neural networks do not have much to do with biological brains, which is itself an interesting discussion, the very act of trying to understand how they work, right, is a very useful exercise in the field. And of course, in consequence, long-range consequence, I'm certain it's going to have societal impact.
I think if there's one thing that people from STS, where I come from, science and technology studies, can do very well is think about dystopian or utopian futures. So we often fall into this binary. Often the reality is more complex, but often we tend to do this. And I think there's this one utopian future that we had in mind, I think, with every big technological advance that is sort of out there to sort of make sort of life better for people. So, and I think this is as big as it gets, of course. But there was also, specifically with AI, that there are so many things we do in everyday life which could be automated, which could be reduced, and I think we often also don't see what work these kind of technologies, then in the end produce for, automating these target activities, so to speak, and then we have the spillover activities that emerge from them. And we often tend to just focus on this one thing, and what I would imagine from AI, because it opens so many opportunities, not so much to focus on the outcome, but on the process. So how to get there, and I think we should just care about, involving many perspectives, thinking about also the people that are sitting now somewhere, categorizing images for I don't know how much dollar per week, just to train these big models that we have out there. So I think my utopian future that I would imagine that could also be boosted with the current AI hub to make this more of an equitable, involving, and just inclusive process in the end.
So the transition to this kind of positive future needs to be handled carefully in your opinion.
Responsible. Probably, yeah.
Maybe in the protein field?
I mentioned already, I think that it's scaling up, right? So if you scale up things and you can do more things and faster, can get closer to these, big questions that we were saying at the beginning. But this will happen if we have this responsible use, because I have two concerns. So one concern is that we burn the methodology, because if you start to produce things that are not producing the effect, then from this high popularity of AI, we will go to this low popularity of AI, right? That there is no trust on the methodology, but it's just because it's not used in the right way. And the other thing is the way that we will change the education system. This is, for the university, but also for high school, because AI anyway is entering in our daily life, and what we need is what we were saying before. We need intellectual property. We need the smart ideas. So if we automatize everything and we kill the ideas, then and if this is not at the beginning from the education of children to how to use these fast tools, but to keep the creativity, then I don't think that also this speed up will be progressive. Mm-hmm. So I think that the answer were more pessimistic, optimistic. But I think that the trend is positive, but it depends how we adapt to this trend.
So how would you say, assume all these challenges are solved?
So if you can really scale up everything, of course you can find much easier, more therapeutic solutions. All right, to make a drug now we have 20 years. If you can reduce it, you have more drugs. I mean, this is a reality. This already happened. We do things much faster than before. So if we go back, we say that technology helps. I mean, it's not being against technology. It's just that this technology is automatizing so much that we have also to care about ideas and our creativity behind this technology.
Thank you very much. Maybe in the clinical field.
It's such a big question. It's really hard to answer. But well, what I think is one of the advantages, or the most important advantages of the new technologies are that they can grasp complexity. And coming from the medical field and from the applied medical field, where we really try to make our efforts to help the patients and to improve treatment, it already feels that the new technologies allow bringing things together. They allow to integrate. So it seems that a lot of the knowledge that was generated the decades before now is being enabled to get to the patient. So that's one of the positive perspectives. Another, I'm purely positive. I'm 100% positive. Another level of complexity, or another example of why complexity should improve health, is hospital management. It's a super complex process, and it needs to be more efficient because the resources are getting less and less. So the resources money-wise, the resources, human resources. So you need to be more efficient. And also think that the new technologies can really improve this. And there are many examples or many visions. Also the surgery, the operation theater. It's now the data are collected that will allow in the future to optimize processes during a surgery, which is it's a vision, and nobody knows if it really will ever work. But even if it stays a vision, there will be some improvement, and there are so many more examples I could think of, or which are underway. And I mean, I think the whole thing is now getting off. And if we think in 10 or 20 years later, there will be, of course, it's a hype cycle, but it's something of that will remain for sure.
Yeah, thanks very much to our speakers today. So I would like to stress that for remaining questions from the audience here and further interest in the topics discussed, we invite you to join the second part of the panel that will happen after a rough 15-minute break. And there we will dive into the audience questions that have been collected, hopefully, but if not, you can still scan this QR code and submit some questions there. Again?
You can vote the question.
Yeah, and I should mention that you can also upvote questions from others, so not everyone has to restate the same question. If you see interesting questions, feel free to raise their priority. Now, let's take a 15-minute break. We have some drinks and small snacks prepared outside this room, and feel free to enjoy these. Thank you all, and see you shortly after the break. So first, from a person called Yeri, maybe quickly raise your hand, thanks. Your question is, you discussed how biological neural networks process signals. Could you elaborate on the learning algorithms they use compared to AI's backpropagation? Can Hebbian learning replace backpropagation, or is this still open? Any good resources to explore further for Professor Mlynarski?
Yeah.
Can you?
Thank you.
Thanks. So it's a somehow quite technical and quite interesting, very interesting question, right? There is something surprising, right? That all of these methods, right, local learning rules, such as Hebbian learning, which can be used to train, in some extent, certain kinds of simple systems, for instance, perceptrons, right, singular perceptrons, or were inspired, again, right, by biology, but right now, we are in this very interesting point where we don't even know if what we thought is happening in neurons during learning and what observations that led to the development of those local learning rules are actually what is happening, right? Are actually the way biological neurons are learning, right? So this interesting duality, right? That what inspired development of technology was our interpretation of the data at a certain point, and technology developed and went its own way, right, with let's say, Hebbian learning, but our interpretation, understanding of biology is slightly changing, like, oh, whoopsie, right? It's kind of a different story, right? So that's one part of the answer. And then the other part of the answer is, of course, discussions about these methods of optimizing neural networks, which are not biologically plausible, we often said, right? So they cannot be implemented locally, or what we assume, they cannot be implemented using ways which we assume at the current understanding of the brain is using. For example, gradient descent, right, with backpropagation of errors. I'm not carefully following that discussion in the field because it kind of assumes that we are smarter than biology. I don't like the term biologically plausible because it means I know what biology can and cannot do, right? And the first rule of evolution is that we are all more stupid than the evolution itself, right? So in that sense, I'm not diving into, but in a very general sense, certainly, right, biological systems learn much more efficiently and faster and robustly than whichever most developed system we have. So in that abstract sense, I'm optimist, right, that we have things to learn, right? But how, time will tell. Yeah.
Did that answer the question?
Yes.
Okay, so second question also to Professor Mlynarski. What are your thoughts on using quantum computing to simulate and better understand the functioning of the human brain? This idea has been gaining traction lately and was initially theorized by physicist Roger Penrose. Would this be the revolutionary idea? From Moers, if you want to comment, also please raise your hand.
Yeah, so that's interesting. Roger Penrose is a titan of intellect, right? And as every titan of intellect, he's permitted to have intellectual hobbies, right? I met myself the Emperor's New Mind. I read myself the other I forget the title of his first book, right? It's a fantastic scientific books, which in addition to introducing somewhat quirky and very unorthodox ideas, right, are themselves perfect, general science exposition to certain basic ideas in physics. That said, I do not think that what stands between us and understanding how nervous systems work, human or otherwise, right, on whichever scale, is the need of invoking quantum phenomena, right? I think that there is so much that we do not understand at the level of at the very macroscopic level, even behavior, and we don't even know what is there to understand, so that, even if there are some effects intrinsic to that at the quantum level, we still have a long way to go. That said, that this belongs to this side of big ideas, right? Cluster, right? Big, great ideas. But that said, there is a lot of research in asking whether biology can operate at a quantum level, and there are phenomena in our brains, also our brains right now, which do not maybe contribute directly to consciousness, but are ways in which biology learns to exploit quantum phenomena. For example, in your retina, right? When it's completely your eyes, when they're completely adapted to darkness, They can count individual photons, right? You can sense change over the order of 20 photons, which is a physical limit. And that is already, biology came up with a way of catching and trapping photons with proteins, right? Which you think about the difference of scales, that's a fascinating phenomenon, right? So maybe not consciousness, right, but the corner between quantum phenomena and biology is definitely a productive and a fertile field of research.
The next question to the entire panel in this case. Based on your discussions on the current trends in applied research, do you see the rise of these fast, novel, and widespread technologies as a risk to the reflexive practices that most scientists would say make the essence of research itself? So, as that is kind of a complex question, I would say, could you ask this in this case, maybe elaborate further and maybe make it more precise on why you asked that?
Yeah, I think like it was mostly inspired by all the discussion that we're having about basically the risk of AI on like a methodological level and the way we do science, and like how do you see specifically like the rise of AI as this type of like where you talk about like we produce a lot of outcomes, but there's not like a focus on the methods. And it's like do you think, what do you think could happen with like scientific research in general if that continues, and how can we avoid that, for example?
To the entire panel in this case.
I mean, I blabbed already for a few minutes, but I'm itching to answer that. So is it okay if I start? Sorry, yeah. You know, I think it's a fascinating question, right? And it's exactly, you know, you are nailing the zeitgeist somehow right now, right? Because we are speaking about German words, right? Because I think we're at a moment where there is a certain kind of, I read this somewhere, schism in science, right? Split. So far, natural science was bringing together understanding of phenomena and capacity to predict phenomena, right? If I understood it, I can predict. Right now we see that they are somewhat dissociating. Maybe not completely, but to a large extent. We can build systems which predict but do not generate understanding, and conversely we can try to build our understanding of certain phenomena without improving our prediction, right? And now I think it will be somewhat boring to commit that there is one way of doing science, right? I think that will add to the plurality, but we should be aware of the fact that there are these different paths, right? And your personal, aesthetical, and ethical, and whatever other choice should be which one of these values you consider more, right? And you know, there is a lot of discussion to be had here. Someone else wants to?
I can just go on. Yeah, that resonates a lot. I think also with what I'm now trying to say. So I think you often differentiate in terms of, you know, there is this differentiation by Kahneman, you know, thinking fast, thinking slow. There's also some talk about, you know, slow science and movements to really make more room for these more immersive sort of kind of methods and not reproducing, you know, certain methods at a fast pace, how it would happen in machine learning, just to, you know, break the next benchmark and to make these models more accurate, etc., etc. So that has happened, I think, throughout the last years. Just to maybe an anecdote, when I was at a robotics conference, people talked to me, and we are in an age where many fields are actually sort of declining or dying. If you think about fields like, so talking as the social scientist here, so if you think about fields like philosophy, etc., people hardly get funded nowadays, you know, to work on classical social theory. So that's a big issue from the domain where I come from. And on the total opposite, you have something like robotics, which is completely exploding. And you know, people also, reviewers at robotics conferences are actually now worried about, okay, what is this next generation of roboticists that is coming there, and what are they actually focusing on? And they are actually worried about, you know, doing this science in this fast mode, you know, reproducing, validating, or not even validating, reproducing certain methods, creating similar outcomes, but not raising these fundamental questions and these fundamental theoretical, or bringing new fundamental theoretical insights into being. So this is definitely their concern. So this question, I think, is really spot on for the domain of robotics at the moment.
Yes, I add a small piece. I mean, I think that we are also, I mean, the way to make science is also dynamic, right? So we have also to adapt to, I mean, it was not the same 5 years ago, it was not the same 10 years ago. But the idea is to bring some knowledge to humanity, this piece of knowledge, and then you can use any way, right? So it can be understanding, can be predicting, it can be, discovery, it can be, having discovery without, for years we had empirical discoveries.
And by using sequencing technology, we now can say, okay, there's a rearrangement between chromosome, this part, and another chromosome, another part. And this leads to a sequence change, and then leads to a protein change, and then it comes to a phenotypic change. And this all we can do in such an extent, in such a complete way from DNA sequencing data by using computational methods. But there is a fundamental understanding of what chromosomes are, what nuclei are, and what cells are. And this is where I see, or where I would put a red flag. If you only have this explanatory layer without the fundamentals, at some point, if you think about 10 years further, no one will be able anymore to explain you what a chromosome is, what the mechanisms are that lead to breakage of the chromosomes and all the effects downstream. What then? Is this, and of course, it will always come back to the fundamentals, because when you at some point you will find a new phenomenon, and then you will need someone who brings the ties together and will come up with an explanatory model. And this is something that is really a problem when it comes to wet lab. There is many wet labs dying because they don't get funding in the moment, because there is so many explanations that can be delivered by in silico and computational biology, biological methodology. But this will not pay out, because you realize in 5 or 10 years time that we will need this layer of explanation. And this is something that has to be tackled, realized, recognized, and communicated by politics again. This is really a serious topic.
The next question is about bias by Emmanuel, if you would like to state your question. If not, I can phrase it for you. So, which biases arise in your respective research field when using ML, so machine learning models? If so, how can these be addressed, also to the entire panel?
What kind of biases? Well, I mean, so when I think of the medical field, one of the most important biases comes probably by population sort of biases. So when you have your models developed in populations that do not reflect our population, then we get to a bias, and the model will not at least provide the optimal prediction. Again, I could make up a political topic here, which goes down to data protection and data protection politics and policies. If we in Germany don't find a way of a more pragmatic way of data protection, we will use models that will be developed somewhere else in the world, and which will not provide the optimal predictions and the optimal performance that we would like to have.
This is, there are bias everywhere, right? And it depends on the data and the funding. So, there are thousands of rare diseases in my area, so in the drug design area, where you don't have funding, you don't have industry investment, you don't know anything, and no one is working on them. For example, for the food context, I mean, the biggest database that is used for olfaction is about odorants molecules that are called key food odorants, and are the molecules that are important for the smell of food in the Eastern culture, right? So, you know, and we completely exclude the other foods. I mean, it depends where the data are generated, right? And this depends on, I think, two factors. One, you know, strategically, politically, but the other is also the tendency to go to where is fashion to go, right? And this is also own responsibility at the end. I mean, because I mean, if it's more trendy to work on something instead on something else, then you get more grant and more collaborations, and it's also a way to see how science can be the management of science. But this is maybe a bigger topic. But I think that there are bias everywhere, and it's important to know it.
Have to think a bit further.
So no, we can talk about, there are many meanings of the word bias, right? There are societal biases, there are the economical biases, right? But thinking more about technical meaning of the word bias, right? The funnel of data processing, AI intelligence, right? So a bias meaning being attracted towards some sort of set of solutions, right? The space of all possible solutions to problems. For some reason, I'm being attracted more, right? I'm more tilted towards, right? Biased towards certain kinds of solutions. For example, in estimation problems, right? And there are many examples of those biased systems which challenge our sense of ethics, right? One famous one, which made circles on the internet, a super resolution system which probed with picture of Barack Obama with low resolution, right? Painted a completely white person, right? Because was biased towards the training data set, right? But there is a positive in statistics we also have biased estimators, but there's a positive sense of the word bias which comes up in our work when we study sensory systems, vision, audition, smell, whatnot. You can show statistically that in order to solve problems with partial knowledge, you have to know something about the distribution of quantities you're trying to estimate in the environment you evolve in, right? There's no general solution. You're solving only in the context to which you adapt it. And for biological organisms, this means that they are biased naturally towards statistics and irregularities they encounter in their natural environments. Right? So we study properties of natural environments, natural sounds, natural images. We can predict something about biases in the nervous system, right? About the structures they will prefer. And that was that's a very fruitful and rigorous and interesting direction of research in computational neuroscience, also with an interest of our group. And it turns out that a lot of properties of sensory system, vision, hearing, also perception in humans and animals, can be explained as a bias toward their ecological niche. So there's also a positive meaning of that word. I apologize. I have to nip out. So apologies to fellow panelists and to everybody. Many thanks to the organizers, and I hope it's going to be fun afterwards. Yeah, thanks.
Yeah. Just in case you were wondering, we specifically prioritized the questions here. So all the most pressing questions have been asked already. Okay. Maybe the bias question. Continue, please.
Yeah. So actually, what Wiktor said just resonates a lot. I think what I always, if I hear the term bias, then I always ask, what do you mean with it? Because from my perspective, or from I think many colleagues would also agree that sort of bias is not a bug, it's a feature, right? So it's not about, because often if we talk about bias, we assume there is data, here's the model, and if we train the model with the right data that is non-biased, then we can eliminate bias, or at least create some accurate sort of prediction machine that is unbiased. But I think it's just important to keep in mind with all these topics that these things are trained with certain data that emerges from a certain context, from a certain socio-cultural or socio-political context, and this is always something to keep in mind. So yeah, bias is a feature, not a bug in that sense.
The next question is about AI in the gastronomic field, targeted at Professor Pizio. If you would like to state it yourself, please raise your hand. Yes?
Okay, thank you. I wanted to ask, this was my question that I wanted to ask you and also Mr. Braun about AI in the gastronomic field. I feel like since many years it's been a very controversial topic how we develop technologies in this field. It's, I don't know, I remember since high school we talked about GMOs and how it's so problematic. I wanted to ask you how you would approach presenting the topic of AI in the gastronomic field to the wide public. Because if we want to apply these technologies, which are very interesting and good, we have to make them available for a wider range of people.
Yes, so I mentioned this in the last slide because this is a field, right? So there are, this is a growing field. There are companies and scientists trying to make this health Alexa, right? This is one prototype. And I mean, for everything you can have to, and it can be technological optimistic, everything will work, everything is nice, or pessimistic, and usually the truth is in the middle. When we talk about food, gastronomy, there is AI cultural component. So we don't eat because we need to eat. I mean, this is something that an old lady told me when I was in my town last summer. She said, we don't eat anymore because we are hungry, right? So, I mean, we eat also what we are, right? So we eat what gives us emotion, what we remember from our childhood. I mean, taste and eating and gastronomy is culture in food, right? And this means that we have to incorporate cultural data and not only food chemistry, right? And this is the first challenge, how to incorporate this cultural data. And there is, of course, a own field that is this tick data. So what makes sense, not in terms of big data, but what makes sense in the context. So I incorporated this cultural data. The other thing is the acceptance or something that is generated by the computer. And this is, again, another cultural thing. I mean, if you would like to, eating something that was done by a 5-star chef will be similarly accepted like a computer recipe. And there are many components when we go to something that is cultural and it's not only chemical data, right? And this is why it is more complex than just the language, in my opinion. But I know there are chefs that now do this with AI, for example, right? So I mean, it's not an idea; it's a proposal. So there are AI recipe. There are AI app that you can picture and see which are the components and how you can mix. I mean, there is a real direction, but this is very multidisciplinary. This is not only about the content of food. I don't know if this answered your question.
I think it was also for Mr. Braun.
Yeah, so being someone that comes from a chair that prides itself to be called Science and Technology Policy, I would also maybe suggest to make these things maybe also political questions in the end. So I think we often so I completely agree, I think, with the points you made. I think we could basically build on that to also offer choices. So I think there's, in STS, often this case of the acidumar case, with the mRNA research back in the days in the 70s, where there was this moratorium that this research should be halted, and there were good reasons for it, but this decision was made by the scientific community itself, and not so much a public process of engagement. And this is also something we see in the many research projects that we have, because you often talk then about acceptance or irrationality, that there are these concerns about also AI misconceptions, for example. But if you talk to people, oftentimes it's more about their livelihood, or about how these people are embedded in a social system, which they now fear might be lost, how their livelihood changes basically. And there to also say, open the process up. So just what we did in the Responsible Robotics project, for example, to open rooms where people can also, where you show this is the research, what do you think about it? For example, a health Alexa, in the end, how do people think about this? And I think there are, for one, many interesting insights to be gained. And also it's just one means to avoid these this feeling or this sort of the concerns maybe in the end that often revolve about different things than just not knowing the science right, or just not knowing what is actually happening.
So yes. So the next question was about for Kristian Unger here, and about the survival distribution graph you showed in your presentation. Would you like to say that question yourself from the audience? Okay, if not, I will phrase it. So the question here is, do you think that this distribution you showed about the two different survival probability, two different curves basically, are biased towards survival of the most terminally ill patients because they are more likely to accept to participate in this study, or was it accounted for when you generated or acquired this data with the patients that you normalize basically if there were enough less ill versus ill patients then?
Okay, so the participation of the patients in the study for certain is not biased by the prognosis these patients have. So at the point they entered the study, so that was quite before, a few weeks before the treatment started. Actually, there's no intervention with taking part in the study, so there's no decision point whether there is an advantage or disadvantage by taking part in the study. There's only the decision point whether they lead to a potential benefit in the future. So and the percentage of patients taking part is over 90% anyways, throughout all prognostic groups, and as I said, you don't know beforehand to which prognostic group you belong to, so I don't think that there is a bias at all.
I think that last statement was very insightful here, because obviously, if you look at the data after the fact, you might think about is there bias in it, but when you acquire the data, you don't always know, or most often you don't know the outcome. So it's probably not biased in this kind of position.
Well, yeah, actually, so we have two different setups. So we have a prospective and a retrospective setup. When we do the retrospective setup, we would have the potential to select for differential prognosis. But at the time the samples were taken, so when the patients were operated or biopsied, nobody knew what the prognosis will be. So
thanks for that deeper explanation here. The next question is about molecular dynamics. Would you like to state it yourself? It's also to Professor Pizio. Molecular dynamics compared to machine learning to predict it instead.
So basically, there is a huge.
Can you hear me?
Yes.
Okay.
So there is a huge revolution right now on machine learning and molecular dynamics in general. So normally we as biologists think of the structures as completely static because it's what is presented in the different lectures and all these kind of things. And basically right now with this molecular dynamics, we are seeing a huge amount of information, and there is new people designing this machine learning approach to understand or trying to simulate the simulation, so go to the different local minima area. So what I would like to ask you is, what is your approach to this new area of the molecular dynamics? And if you think that there are ways of having better database or curate better the data of this molecular dynamics, because you know it's really dependent on the temperature that you use or all these parameters that you actually change a lot when you are doing these simulations.
Thank you for the question. I mean, proteins are not static, they move and they move a lot. They move so much that it's difficult to sample with molecular dynamics. So even now, the computational power is increasing. It's much easier to do MD. It's always not complete for the complete conformational landscape. So there are two questions. So one question is if machine learning can speed up the process. So AlphaFold, I mentioned AlphaFold. We are talking AlphaFold as is the most popular, but together with AlphaFold there were many other AI tools, and after AlphaFold there are many derivatives, and including this conformational sampling. So there is this AlphaFlow. I mean, there are at the moment not really it's difficult to compare and to see which is this possibility to generate different conformations from machine learning. But if you, in general, it depends what you want to do, right? So if you want to generate different conformations without not a complete sampling, any start point can be a different start point from MD. If you're looking at a specific event, you have to follow the physics, right? So it really depends, but with static structure and with very low resolution static structure, there were many progress, right? So if you can improve it, I'm usually, if you ask my personal approach, I'm usually open to increase the portfolio of technique that you can use, since the problem is so complex. But it's also, I'm very happy that I received this question, because if we increase the possibility, we also have to be aware of this aspect. And actually, there are growing database on simulation data. So we work on one of these databases that is specific for G protein coupled receptors, but now there is a European project for this database. So it will be possible then to increase also this area. Thank you.
And the final question we have right now on the list is for Professor Unger about the size of the data sets, I assume. I think you might want to ask the question.
Yeah.
Thanks.
So the backstory of this question is that I was working on a research project. They had this protocol and wanted to optimize it with AI while taking different pictures and different time points of the project, and then seeing if it would be successful, so to say. You talked about feeding the AI some histological pictures. So I was interested in how big of the data set must be for the AI to give a reliable prediction of these prognoses that you talked about. Around how many pictures?
So this is one of the most important questions when it comes to study design, of course. When it comes to conceptuating a study, also against when it comes to applying for funding, for instance. I mean, you have to come up with a target sample size, and but the answer is you can't tell beforehand. You need to try out, at least in a pilot. The thing is, for instance, so one of my students, Roche, he had to leave, unfortunately, so he could have given you even more informed answer on that. He works on the histology images. So we are using a foundation model. This foundation model was trained on more than 40,000 of these kind of images, and can give you a representation of your actual histological image. And using this foundation model to predict obvious factors such as anatomic subsites. So when we look at head and neck cancer, the cancer could be in your mouth, it could be somewhere in your neck, it could be in different places. With only around 250 images, we can, with the accuracy of around 86%, we can predict the anatomic location. So it also depends on how subtle the effect is you want to predict. In the case of having already a pre-trained foundation model, you don't need tens of thousands of images to get to a successful prediction model. But still, it could be that the information you're looking for or the information you need to do your prediction is just not in there. And this then comes back to plausibility. A lot, I think, when it comes to study design. So if you want to predict whatever the intellectual properties of a patient from a histological image, this will taken from the breast. This will not be successful because the information will not be in this image. So you have to understand the biology and the pathology, and in order to get to a plausibility, to a plausible way of thinking, is the information likely to be in your data or not? This is, I'm answering very cloudy. I know that this is not a concrete answer, but it's not that like in power analysis, where you can say, okay, I need this number of cases for this effect size, that I will find an effect with 80%. You have to try out and find out. But I think the whole power was very much boosted, or is very much boosted, by foundation models, and it will get even more improved by new foundation models.
Thank you.
So yes, with that we are gonna wrap up the question and answer session. If the speakers will still have time afterwards, at least there's beer outside, so anyone interested can have one. You may ask them additional questions that you hadn't had the chance before. But for the panel discussion here, I would want to thank our panelists for sharing their expertise and their perspective. And a big thank you to everyone from the audience here for attending and voicing your questions here. Thank you very much. So as you can see, but for the rest of you, feel free to
thank you so much. Yeah, one of our team members actually did it herself.
And,
so.
But otherwise it is nice even and get home safely. This was very insightful. We brought a lot of different perspectives into this discussion. Very amazing, actually.
That was really good.
I mean, I enjoyed very much also your organization.
We had the perspective of the
Very well done.
It's a perfect job.
Yeah.
No worries.
One okay? Is this hidden?
Yeah. Okay. 3, 2, 1. Another one. Ready.
Thank you.